So, good evening, everyone, and welcome to another edition of Pint of Science, coming to you live from the University of Bath. I'm Ghatora, and I'm a second-year PhD in the Department of Biology and Biochemistry. And tonight, I have the privilege of being your host for the first of a theme of talks for Tech Me Out called Creating and Detecting Life Using Tech. I hope you've got your seatbelts on for an exciting evening of talks. And please make sure you check out the Pint of Science website for a series of talks that will be happening over the next 4 days. I do encourage you to please comment where you're coming in from, where you're viewing from. Sorry, we've already got quite a few comments joining in from people joining in from Scotland and Bath. I thought I'd first like to get things started with a quick question for you, our wonderful audience. As this is Pint of Science, I thought my first question for you all would be, whose scientific work, it could be in the last 10 years or the last 100 years or even beyond that, whose scientific work do you think has had the biggest impact on human society and why? As an example to get you started, my biggest inspiration is an American biochemist, and her name is Jennifer Doudna. If you're not familiar with her name, Jennifer is one of the pioneering scientists who was responsible for the development of CRISPR-Cas9 gene editing technology, which has a variety of applications in medical therapeutics and even fundamental genetics in both bacteria and humans. So please do let us know who your favorite scientist is, who you think has made the biggest impact in science on humankind. For now, I'd like to get started with our first speaker for tonight. So our first speaker tonight is a fellow PhD student from the Department of Biochemistry. Her name is Victoria Riddell. She hails from the labs of Stephen Bagby, Pedro Estrella, and Nia McHugh. And tonight, she's going to be taking us on a journey of electronic devices to help for the early detection of autoimmune diseases. It's nice to see you, Victoria. How are you?
I'm very good. Shame we're not in a pub, but it's nice to be here. Thank you for the introduction.
I could do with a pint. Well, I'm very, very excited to hear you talk. So without further ado, I would like you to take it away.
Perfect. Thank you very much. So today, to kick off this theme, I'm going to talk a bit about my research in creating a biosensor for autoimmunity diagnosis. Now, I know some of you might not be experts in autoimmunity or biosensors, but don't worry, I'm going to go from very basic and talk about, in general, technology in research and how it's really advanced in the last couple of years. And in particular, I want to talk about technology in medicine, because I think that this is a really interesting area. And if research is going the way it's going, I think we're going to end up with personalized medicine in the future. So to kick off, the first question is really going to be, what is a biosensor? And for me, I would describe it as a perfect mix of biology and technology. And a really simple way to think about a biosensor is that it is something which uses biology to detect something else. Very, very basic, but it covers exactly what a biosensor is. And the next question that you might think is, well, detect what? Something is a very vague word. What is it that we want to detect? And this can actually pretty much be anything. It can be incredibly varied. I did say I want to talk mostly about medical biosensors, and I've got an example here on this slide of a glucose sensor, which is a perfect example of a medical biosensor. But you can also get biosensors, for example, to detect, to look after crops and make sure that they're in ideal growing situations or measure pesticides. There's just more and more sensors being used in everyday life. And it is quite an exciting research topic, so there's lots of research happening, but not just within the lab. I think in everyday life, we're seeing more and more mixtures of biology and technology, and more biosensors. So an example of this is this Dexcom watch. So this is a biosensor which has been designed for patients with diabetes. It does real-time monitoring of their glucose level. I'm not sure how accurate it really is, but it's definitely an example of where the science is going. And it's really exciting that we're starting to see a lot of biosensors more in real life and not just in laboratory conditions. But my question is, if we've got these biosensors and they provide us with a lot of data, how can we use this data to answer some quite interesting and potentially difficult medical questions? So this brings me to autoimmunity, which is the main topic that I research. And you might be thinking, of all the medical questions, why autoimmunity? I've studied autoimmunity for a couple of years now. I worked on it in my master's as well as throughout my PhD. And one of the main issues with autoimmune conditions is that they're often diagnosed quite late, and they're quite difficult to diagnose. And the reason for this is often the condition has started and there's biology going around in the background, but there's no symptoms until quite late on in the condition. And this means because there's no routine tests currently, you're not going to recognize an autoimmune condition and quite often not get diagnosed until it's too late. And the treatment then becomes about managing pain and managing symptoms, but a lot less about how you actually stop the condition from progressing in the first place. So it is really complicated and it is quite difficult to diagnose. Now, if you bear with me for a second, I just want to explain a bit of the science behind autoimmunity, and then I will bring us back to biosensors to explain how this might help with us creating a biosensor. So in this image here, I have pathogens or antibodies. Let's say that this yellow blob here is our pathogen. And what normally happens when you have a pathogen in your body is an antibody will recognize this pathogen. It will then produce more antibodies which are specific for this pathogen so that the body can clear these away and remove the pathogens. And that's great when it works. Incidentally, it's also how some vaccines work when you provide the body with a little bit of a pathogen so they can create lots of specific antibodies. But in autoimmunity, this pathogen, or what the body thinks is a pathogen, is actually just an essential part of your body, an essential protein, which is working away, doing its business, and all of a sudden an antibody decides that it's a pathogen and begins to produce these autoantibodies, which antibodies specific to proteins that are just happy, healthy proteins. And we can now call these proteins autoantigens. And the problem is that when this happens, this is going to kick off a cascade of reactions. You're removing this essential protein, and that's how most autoimmune conditions are kick-started. My logic in detecting this is that autoantibodies are therefore essentially marker for autoimmune diseases. If your body is producing lots of a specific antibody towards a healthy protein, then that autoantibody shouldn't be there. If it is there, then that's a very good indicator and marker of disease. So I want to make a sensor where I essentially use these healthy autoantigens as bait to detect autoantibodies. So the logic would be I would put some common autoantigens onto an electrode surface to turn it into an electrical circuit. Now, I know I said autoimmune conditions are quite difficult to diagnose, and it's often really difficult to know which antigens and proteins are going to be targeted, but we do know in certain conditions which are likely to be targeted, and we can take into account risk factors such as genetics and smoking. So we could create a panel of proteins or autoantigens which are likely to be autoantigens and use them as bait. And if we add a patient sample, which has, it could just be a blood sample, onto this sensor, and there is autoantibodies in that patient sample, then this is going to complete the electrical circuit and we're going to get a measurement. So we're essentially checking if these autoantibodies are there or not. I think this sounds really cool, and so far the research is working out, but I don't want everyone to get super excited about this amazing biosensor. this is actually more what it looks like at the moment. So in the corner we have, that is my gold electrodes, and I put this into a little electrical circuit. So on that gold electrode, I will put some proteins. I then put patient sample on top of that, and I put it into an electrical circuit, and I can take some readings. And this is how my research is looking at the moment. But I think it's really exciting. I think especially if we can create different panels for different autoimmune conditions. This is very generally a proof of concept for autoimmune conditions which are caused by autoantibodies. It definitely will lead to an earlier diagnosis, and hopefully that means more personalised medicine. So I'd like to thank you all for listening. And of course, I'm part of the University of Bath, but I'm also supported by the Bath Institute of Rheumatic Diseases, and I'm part of the Centre for Biosensors, Bioelectronics and Biodevices. Thank you all very much.
Thank you so much, Victoria, for that wonderful talk. That was absolutely incredible, especially since it's a topic that also relates to my field of research, investigating biosensors, but for the environment. Thank you once again, and we look forward to having you at the end to answer some of the questions. It's also great to see some of the comments that are flooding in from examples of scientists who have helped make a massive contribution to human society. So we've got one example of Dr. Caitlin Carrico, whose fundamental work in mRNA vaccines has helped pave the development for some of the COVID vaccines by Pfizer and BioNTech. And we've also got Emmanuel Charpentier, who is also one of the co-founders of the CRISPR technology, along with Jennifer Doudna. Thanks for all the questions that are coming in. Please keep them coming in. If you've got any questions for Victoria, put them in the chat and we'll get to them at the end for the Q&A. Once again, thanks, Victoria. And for now, we've got something slightly different for you. We've got a bit of entertainment, and it's in the form of a chat show from our guests, Peter Harper and Tara Johnson. Peter is a veteran environmentalist who will be interviewing Tara, who should be playing the role of a water molecule from Wales named Hydra. I'm just as eager as you are all for this chat show, so buckle up, get your drinks ready, and please welcome to the screens Peter and Hydra.
Hydra, a water molecule. She joins us now from Wales, where she has, of course, been very busy.
I have been very busy, yes. I just want to clear up one thing before we start, if that's okay, right?
Yes.
I'm not common, I'm abundant.
Yes, well, of course, yes. It's very nice to see you, Hydra, my dear. I'm sorry about these awkward online arrangements. Are you quite comfortable where you are? We wouldn't want you to evaporate or freeze.
Oh, don't you worry, Cariad, it's all water. I've done my time as ice and vapour. I don't suppose you know what a phase change is like. It's the weirdest feeling.
Yes, I can imagine. Well, perhaps I can't. But which phase do you like best?
Oh, liquid, for sure. It's all dancing, you know, like a Kaylee. Look at these beautiful elastic hydrogen bonds. We're polar, see? Like people, we have tops and bottoms. But with us, our tops are attracted to bottoms.
The mind boggles. But aren't these bonds rather fleeting? I mean, no, you don't have permanent relationships. You know what I mean?
Relationships? I haven't had a permanent relationship for 50 million years. The bonds come and go, and thousands of times every second.
Well, what about your domestic arrangements? I mean, you know, the three of you together, hydrogen and oxygen.
Well, we rub along okay, share electrons and stuff. We just need to keep the hydrogens apart, stop them squabbling.
Really? How far apart?
Well, we found that about 105 degrees is about right. Mine are called Ceres and Mofamwy. We do feel at home in Wales. There's something about it that attracts water molecules, and our emergent property of wetness, well, just emerges.
Nghymru yn wlyb, ond gwlad beirdd a chantorion.
I'm glad we cleared that one up. Now, you're famous for being a brilliant solvent.
Oh, thanks. But it is sometimes a bit irritating to be regarded as just a solvent. I have got to say, though, distilled water is rather boring. Nothing to gossip about. It's just us. Solutes make life interesting, especially the organic ones.
Oh, like in bodily fluids, blood and things like that.
Yeah, and don't forget urine, saliva, sweat, phlegm,
What?
snot,
Hydra?
mucus.
Really?
Semen.
Oh, no. Thank you.
Oh.
Yes, thank you, Hydra dear. Perhaps can we change the subject to inorganic dissolved molecules?
Well, the ones I like best are our cousins, the oxygens. They keep order. Funny, they always go round in pairs, like cops. Like they do. Sweet as pie most of the time, but give them a dodgy molecule and they beat the shit out of them. No mercy.
Really?
Molecular piranhas.
Shocking. Speaking of which, what do you think about electrolysis?
Well, we don't mind being split up for a bit. The whole point is to get the hydrogen and combust it with oxygen to make water again. It's a bit like reincarnation. You disappear in one form and reappear in another.
Well, I'm very glad to hear that you're easy with electrolysis. Now, there's something else we'd really like to know. Where did you all come from?
You know what? I'm not entirely sure. We've got two stories. One about coming from inside the Earth, the other from the comets. Not sure which I like best, but the results are good, don't you think?
Well, it's hard to say. But look, we learned a rhyme about this in the infants.
Jack Tar Sailorman, can you tell me how much water there is in the sea?
Yes, miss. Yes, miss. Certainly. There's just as much as there ought to be.
Oh, that one. Actually, that's one of my favourites.
Well, is it right? I mean, is there just as much as there ought to be?
Well, here's the thing. The universe works in orders of magnitude, not piddling percentages. It's 35 orders of magnitude down to the Planck length and 26 to the edge of the universe. If there was just 1 order of magnitude less water on the Earth, you'd only have a few lakes, look. If there were just 1 order of magnitude more, you'd only have a few islands poking out of the world ocean. So that old sailor man was right. Pretty good balance, eh? Plenty of land, plenty of sea. We nailed it.
Some people would say that that had something to do with Gaia.
Gaia! She gets all the bloody credit, but she couldn't manage it without us, could she? Just look at Mars and Venus. And what's that they say in Bath?
Ariston men
That's
idor.
all Greek to me. What does it mean?
Water is best. And don't you forget it.
Okay, you've convinced me. Now, would you care to tell us something of your adventures?
Oh, yes. I really have done something with my life, haven't I? Of course, most of the time is in the ocean. Don't get me started on those chlorine ions. So smelly and frankly, negative. But it's pleasant enough. Plenty to do with all that bonding and stuff. It's a bit of a scrum where we meet the air, but nothing serious. Just surface tension, you know. And sometimes you do find yourself in the open air.
As a vapor molecule? And what's that like?
Oh, I tell you what, if you ever get blown over the land, the views are bloody marvellous. But eventually you come down as rain or snow and have a nice long run back to the sea.
Don't you ever spend time in plants or animals?
Not often, but sometimes. Once I got waylaid by a plant root and found myself rushing up the xylem into the leaf. And do you know what happened then?
No, do tell us.
It was a kind of forced marriage. You can't argue with those monster enzymes. They got 6 of us together and forced us to engage with some of those dreadful CO2 molecules. We lost our oxygens and ended up as glucose. Then they took us somewhere else, like a prison cell it was. But in the end, those oxygen atoms came charging back like the US cavalry, broke up the gang and released us. I never saw so much energy.
Yes, so you don't like the CO2 molecules?
Oh, they're all right, I suppose. Just so full of themselves, thinking they're the big greenhouse gases, when it's us who do the heavy lifting. We're the ones who make the Earth habitable, and let's face it, there's just too many of them.
So they say, yes. Now, what about animals?
Oh, very rare, God bless them. Less than 1% of living matter.
What?
Sorry, I forgot. You're an animal, aren't you? But there's no arguing with the data. I'll tell you this, though. People say that if you drink a mug of tea, chances are that one of the molecules in the drink would have been in Socrates' cup of hemlock. But I was there. I was in that cup. As humans go, Socrates was one of the best. So it was a shame, really, but I had a job to do.
Wow. Finally, Hydra, I'd like to ask you, what do you consider your greatest achievement?
That's easy. The seaside. Oh, I do like to be beside the seaside.
Yes, thank you.
Oh,
yes,
I do like
we do.
to be beside the seaside.
Thank you, Hydra. It's been a pleasure. Ladies and gentlemen, that's all we have time for.
I should like to.
Oh, she does go on. Hydra, my dear, just shut up. Thank you. Next week, ladies and gentlemen, it's Professor Schrödinger and his notorious cat. Wanted, dead or alive.
So thank you, Tara, Hydra, and Peter for that absolutely entertaining sketch. That was really, really enjoyable. If people want to know more about some of the work that Peter Harper does, please check out his website. It's peterharper.org. If you want to be in contact with him and check out some of the cool stuff he's doing. Our next speaker, we're going to be moving out of the Department of Biology and Biochemistry and moving into the Department of Physics. He is a principal investigator who joined the university in 2018, and his research focuses on the theory of soft materials, including mechanical metamaterials, active matter, topological states, and polymer physics. His name is Anton Souslov, and I'd like to welcome him all to your screen. And so tonight, Anton will be talking about some of the really, really cool developments in designing materials that move themselves and how this field is likely to advance with the development of novel futuristic technologies. So, Anton, it's wonderful to have you here. I hope you're just as excited as I am. Please take it away.
Thank you so much for giving me the opportunity to speak here, and I'm very excited to be here. I've been really enjoying all of the talks, and I am really looking forward to telling you about the work that we do in my group here in Bath Physics on metamaterials, how they live between materials and robots. So in my talk, what I wanted to tell you about are these illustrated by these 4 different pictures, and I'm going to go through all of them in turn. But before that, I wanted to introduce my title, this idea of being between materials and robots, and just go through what exactly I mean. We think of materials as illustrated in this picture on the right. And in a material, what you have is an arrangement of atoms, right? In this case, it's a very advanced material called graphene, where you have a 2-dimensional array of atoms. But all materials like us, around us, look like that. And on the right, what you see is a typical robot, and it looks very different from a material. It's composed of parts that are on a much bigger scale. They are comprised of many, many atoms, and they're put together in a very specific way to perform robot function, to run, to jump, or do whatever else you want the robot to do. But there's an overlap between the two fields that study these different things, right? And everything I'll tell you about is in this overlap. So the first example I will give is robotic swarms. I'll also talk about mechanical metamaterials, active origami, and soft robotics. And in all of those cases, we can think of these things as both materials and study them using ideas from materials science, as well as robots and study them using the science of robotics. And so this area is very exciting because it brings these two different sciences together. So let me go to the first example, and this is the example of a robotic swarm. And what you'll see in the videos here is a swarm. It's the same swarm in both of these videos, but it's been programmed in slightly different ways. The robots have slightly different instructions. And what you see is that as a result of these instructions, they form different shapes. The swarm on the left is slowly making a star shape, whereas the swarm on the right is making the letter K. And the interesting thing here is that the material out of which these shapes are made are not plastic or ink the way we're used to with stars and letters, but instead the material itself is a bunch of robots, right, that you can program in order to reconfigure between different shapes, for example. Of course, there's a lot to be done in this area still, and what you can see is that the length of time that it takes to make these shapes right now is very long, right? It's almost 12 hours. Whereas what we want to go with this as scientists is to do this arrangements in real time, right? For it to take seconds, and that's an area of active research still. Another example I wanted to go through is one that I've worked on myself, and this is an example that might be called an active mechanical metamaterial. Here too, you have little robots running around in some space. In this case, the space is very small and the robots are very small as well. This whole image, when zoomed out, that I'm showing you is actually smaller than a coin. And what you see is that these tiny robots run around inside this maze that we've constructed, and we've constructed this maze with a very specific purpose, right? It's to give this material a very unique property in terms of how it conducts sound. And what you can see in the video here in this illustration of a sound wave in this material, it only goes around in one direction, clockwise around the edge of the sample and not counterclockwise. And it turns out that you need these robots to create that. Next, I wanted to tell you a bit of a different type of material, and this is something that might be familiar to you from playing with paper. The idea is to create origami that can fold itself, right? When we usually think of origami, we think of as a piece of paper, right? And we need to make an origami, we need somebody to fold it, right? So we have someone who's knowledgeable with the design, and they fold this paper, for example, into a boat using a particular pattern. But in this video that I'm showing you, you do not have someone folding this origami. Instead, you have an origami that folds itself. It again folds into a boat, as you'll see in a second here. But instead, the reason that it folds is because there are little motors at the various folds in this sheet, and the motors actuate in a pre-programmed way, so they know when to fold, right? And by reprogramming these motors, you can go from between the different shapes, right? From boats to airplanes, for example. And the reason that this might be useful is because you might want to have structures that build themselves in some way. One example where this is already used a lot is in space, right? When you want to send a satellite into space and give it a solar panel so that it can charge itself. You want the solar panel to be folded up and then deploy itself when it's out in space. And of course, you don't want to send an astronaut to do the unfolding. That's a very complicated task. You want a robot to do the unfolding for you. And similarly, you could imagine doing that here on Earth in areas, for example, affected by disaster. You want to provide shelter for a lot of people very quickly. You do not have the resources to move a lot of people in there to fold tents, but you could imagine tents that unfold themselves by having these motors that deploy them. And this would very quickly provide shelter and safety for a lot of people affected by disaster. So that's one application where this area of robotic materials is going towards. And now let me give you a last example. This last example might be called soft robotics. And what you see in this video playing on the left is one robot arm or leg that is itself created of a structure, right? It's not a typical robot leg that's just composed of a piece of metal. Instead, it's something that's soft and foldable. And the idea here with soft robotics is that you have these structures that are on the scale of the robot itself, right? And by moving these structures around, you can have the robot move. And so that's what I wanted to show you in this video here, a soft robot constructed by having these 5 legs actuated, right? So made to fold and unfold in a particular pattern. And you can see the pattern of actuation here. This controls the direction in which the robot is moving. And so first it moves across the screen, and then it's reprogrammed to start moving away. And the interesting thing about this approach, the soft approach to robotics, is that it doesn't require any rigid metal components. Instead, it's the components are themselves sort of like pieces of origami or pieces of a robotic material themselves. Well, this is what I wanted to tell you about today. I showed you several examples of this exciting science that my lab here at the University of Bath is very much interested in. These examples all sit at the intersection between what we call material science and we call robotics. And one thing that I hope you take away from this talk is that, I've named all of these examples by in different ways: robotic swarms, mechanical metamaterials, active origami, and soft robotics, but they're really all examples of the same sort of science that uses many of the same approaches. So thank you very much for the opportunity to speak here.
Thank you so much, Anton. That was honestly an absolutely incredible talk. We just had a comment come in from Anna, who said she would love the idea of a self-pitching tent. Having been to festivals, I honestly can't stress how much that would be a good idea, having to set up a tent in the middle of downpour. That's super, super cool. Thank you for that. And it's the time of the evening where we like to invite both of our speakers back, and it's time for our question time. So if Victoria, you can come in. Nice to see you again. Firstly, I'd like to thank you both so much for your incredible talks, and thanks for joining. But now it's time for some quick fire questions. So I'm going to start off with Victoria, and it's a question from Sarah Brent. And the question is, do autoantigens largely overlap between different autoimmune diseases, or would you tailor the sensors more towards one type of autoimmunity that could be more likely to occur in a particular person?
Yeah, so some of them can overlap and some of them don't. It can get quite complicated, but we have disease-specific autoantibodies, which won't overlap. And if you have one of those specific autoantibodies, you might have that particular subtype of the autoimmune condition. But we also have what we call associated autoantibodies, and these can occur across multiple autoimmune conditions, but they'll provide us more information as someone who has 2 or 3 particular associated autoantibodies are more likely to go on and develop lung disease alongside their autoimmunity, or are more likely to go on and develop cancer alongside their autoimmunity. So collecting a whole panel of the different antibodies which are present can tell us a lot about an individual person and how their condition might progress.
That's very interesting. Well, thanks for that. The next question is from Insane Cake, and that's directed to Anton. You mentioned origami for the soft robotics, but what are the actual little robots made of?
So in many different cases, you can sort of make the robots from whatever you want, right? In the examples that I showed you, these robots were mostly made out of plastic with some electronics, right? That just makes them cheap and easy to mass produce. So, for example, in origami, it was a plastic sheet with some metallic actuators.
That's super, super cool. And really, really interesting. Back to Victoria. The next question we have is from Simeon Barberinde. I hope I pronounced that right. Simeon says, hi Victoria, your work is absolutely amazing. I'm wondering how robust your approaches are, since we have quite a number of autoimmune diseases, and could there be different kinds of autoantigens?
Yes, so the talk that I presented today, I gave very general information. My particular research is focused on myositis, which is an autoimmune condition which involves inflammation of the muscles. And there's 2 reasons that I work on this. One is that I'm very lucky to be part of a group in pharmacology who work on this condition a lot and have access to hundreds of patient samples, which have already been tested by multiple different means. And the other reason is myositis is quite an interesting condition in that it has specific autoantibodies which are mutually exclusive. So if you have one myositis specific autoantibody, you can say they've got this exact subtype of the conditions.
Okay,
but it is
cool.
And for myositis and other things, you definitely would need to create like a panel for rheumatoid arthritis or a panel for myositis. Okay,
cool. And so if you make that panel, could you use it to monitor the different progression of like a specific autoimmune disease, like from the start where you would be detecting like one type of autoantigen of the like early onset disease, then midway, and then towards late progression?
You definitely could, and there is a lot of ongoing research, which is quite a hot topic in this particular field, about how antibodies themselves might change in their structure as the disease progresses. And if you could monitor those modifications and changes, I can tell you a lot about what's going on.
That's super, super cool. Thanks for that. The next question, well, it's not really a question, it's more of a statement. It's from Chris Thompson, and this one's for Anton. So you've effectively made soft robotic muscles in a way. I sort of want to follow on with a question for myself based on that. And the question is, do you see any sort of application of like these self-assembling materials in potential human therapy? So it might be like if you have muscular degeneration, is there any way you could sort of implant or incorporate cells onto the self-assembling materials to sort of rebuild muscle or improve, or at least improve muscular movement?
Yeah, thank you so much for the question and the comment. I think they're all really relevant to the research area. So I would say that, first of all, a lot of the science of soft robotics and these robotic materials is very much bio-inspired, right? So it means that we kind of look what nature does well with soft robotics, and this could be muscles or even plants, right? Carnivorous plants, for example, also move. And we try to recreate that in the lab with materials that we can, mass produce with, right? Or to create something that goes beyond nature, right? And in a related comment to answer your question, one of the major applications of soft robotics is in sort of things like artificial limbs, right? So therapeutics, right, that allow us to make, essentially body parts out of artificial materials, right, and have the properties that we, like softness associated with body.
That's really cool. And how far do you think we are from attaining? Because I imagine that all that sort of stuff is still in development. It's at very early stage. But how far would you say, sort of along the timescale, are we from having sort of therapies like that to have like artificial limbs incorporated into like the healthcare system? Would you say in the next 10 years, next 20 years, or is it still quite early on?
I would say it's still quite early on. I mean, I would say that, what is currently, right, being used for artificial limb is often, sort of fairly basic soft robotics. But I think that, in terms of using the kind of the full range of fundamental science, right, what we can develop, it's still quite futuristic, right? I think there's a lot more technological and scientific progress to be made here.
Well, that's super. I look forward to seeing how the field is going to advance and see how far we actually are away from that sort of technology. Back to Victoria. So this is a question from Lewis Irvine. Can you detect other diseases like this? So not just like autoimmune diseases. Could you also use the similar sort of platform to detect COVID, for example?
I'm sure there could be. I'm sure there is. There's quite a big team in electrical engineering with some crossover to biology, who everyone works on individual panels. I know there's quite a lot of people, at least 2 or 3 people working on DNA detection. So it is definitely quite a big area of science at the moment. But I'm afraid my specific knowledge is autoimmune diseases, so I can't tell you more than that.
Fair enough. So I've also just got one more question to piggyback off of that. So this is a question for Victoria from Olga Ezirova, if I pronounced that right. And that is, if this early diagnostic you're working on succeeds, is there existing treatment that patients can be matched with that would improve their quality of life or not?
It completely depends on the autoimmune condition, the subtype of the condition, and if they are likely to go on and develop further conditions alongside. So in some cases there might be, in other cases it might be, okay, you're going to get this autoimmune condition, but because we know this now, you're not going to get severe symptoms and we can stop you from getting lung disease, which is quite a common thing that happens alongside myositis. So it comes back to it being personalized medicine, and the more information you have, the earlier, the more you can really tailor a treatment program, rather than saying, one medicine for all, which is not always the case.
Yeah, that's very, very true. Well, I'd like to thank you both. Thanks for answering the questions. It's been absolutely fantastic having you both give your talks. I'd also like to thank Peter and Tara for their wonderful sketch. Please don't forget about the other talks that will be going on throughout the Pint of Science Festival. Tomorrow, we've got talks again from the University of Bath with a theme titled Sun, Skin and Science. They will also be starting from 6 p.m. Tomorrow, and there's another one on Thursday. Next up, for the Tech Me Out theme, we've got talks from Newcastle, titled A Glimpse into the Future, and then we're going to be heading back south to the University of Southampton for their talk titled Trust, Technology and Society. So once again, thank you all for being a wonderful audience. I hope you enjoyed the talk. It's been an absolute privilege to be your host for tonight. So it's goodbye from me, our speakers, and most importantly, the technical team who have been absolutely fundamental in helping keep the show running for you tonight. Thank you, everyone, and good night, and I hope you enjoy the festival.